Exactly. I think that's exactly right. So we're really interested in, I guess, two things, like looking at, on the one hand, structurally similar molecules to each compound and seeing the relationships between what we found and what each of these other molecules do. But then also looking in other tissues, because I think what's very exciting to us is, these compounds are great in psychiatry, but when you have a neuron or an immune cell or a structural cell, something like glia or just stromal cells, together in a unit, that's a feature that's present in tissues throughout the body. And so we think that there might be contexts where these compounds would have great utility to cure aspects of tissue pathology in diseases that we weren't even thinking about because of the mechanisms are generalizable across tissues.
Hmm. So, what are you guys working on to follow this up? What are some of the next steps here?
Yeah, so we're interested in a few things. So the one of the things that we're doing is we really want to understand in the context of, let's say, psychedelic induced plasticity, to what extent in stress, let's say, to what extent is the immune cell component modulating that plasticity? It's a complicated question because we didn't really get at that specifically. We sort of looked at it indirectly. And so we really want to identify if, the time scale or the strength of how immunomodulation would influence the rescue of behavior. Alex Kwan just had a paper come out, I think a few weeks ago in Nature, just, beautiful paper showing how dependent the process is on, I believe, the PFC neurons. And so, but then, how does the immune system fit into that type of thing? I think that's something we really want to work out. The second thing we're doing is related to what I said earlier, which is we really want to understand to what extent these sort of cells and the principles that we find here are applicable to other diseases or other tissues. And so we really are interested in models of inflammation and how these compounds could be useful. And that extends also to looking at how patients or how it modifies inflammation in patients.
Yeah, yeah. And then, at the beginning, you mentioned that, there seem to be certain forms of depression, potentially even the ones that don't respond as well to medications that are tied to neuroinflammation of some kind. What do we know about neuroinflammation and sort of immune system brain crosstalk in human psychopathologies that have to do with stress and depression?
Yeah, so it is complex because depression is so heterogeneous.
Hmm.
Um, but what we do know, for example, if you look at biomarker studies, there have been meta-analyses that showed elevation of plasma cytokines that are enriched in depressed patients relative to controls across multiple studies. To some extent, the changes in these profiles are associated with therapy outcomes. That seems to be also the case. And then there appears to also be, at least looking at some patient-based in situ studies, there seems to be some evidence that there's at least local low-grade innate inflammation in certain brain areas that are important in depression and also in a affect in mood that needs to be followed up on more, I think. So I think we're just kind of scratching the surface at this point, and we really need to dig in to specific mechanisms and stratifying how one mechanism might be good for one patient subset, but not another. So I think we're really just in the early stages.
Hmm. Hmm. And, what do you think about so you did this work with psilocybin, MDMA. On the one hand, they have very different mechanisms, but as we discussed, there's some reason to think that they could have similar effects despite those differences in mechanism. That ties into Gould Dolan's work and how some of these things, despite interacting with different receptors, can sort of converge downstream in different areas to affect things like plasticity or the ability to induce plasticity. Are you guys looking at other drugs that are, broadly speaking, considered psychedelics or psychedelic like? Or do you think these two are worth focusing on for some reason?
Yeah, exactly. So we are absolutely doing that. So we're interested in other structurally related molecules, some of which aren't even Schedule 1 or psychedelics
Hmm.
that are related to, let's say, psilocybin.
Yeah.
Um, and we're also interested in particular in other psychedelics or psychedelic-like compounds that are clinically relevant. An example here is something like ibogaine,
Hmm.
which, if I'm not misstating things, has been shown to be effective for eating disorders and also brain injuries, which carry a lot of brain inflammation.
Hmm.
And so we're interested in these compounds because of kind of these phenotypic properties that they might be relevant in people, and we want to understand a little bit better what they do in animals.
Interesting. And so, for the effect that you saw, where you have these monocytes getting into the meninges, where they can then induce these changes in the brain, but then MDMA and psilocybin can sort of interfere with that and get them out of the meninges. Does that imply that uh, so for MDMA and psilocybin to have that effect on the monocytes in the meninges, that does not require them to cross the blood-brain barrier and go into the brain? Is that right?
Exactly. So this part appears to be where we're thinking of let's say neuroinflammation in a stress context to be distinct from something that would happen in neurodegeneration. And so the barrier is just weak enough where peptides can get through, something like small peptides like cytokines. But it doesn't appear that the cells themselves get through. So there's evidence, for example, in the nucleus accumbens, where there's also this BBB breakdown, where the immune cells are in the vasculature, the monocytes are in the vasculature, but they don't appear to be actually getting in.